If you're taking AP Bio, you already know that evolution is like the glue that holds everything together. But have you ever stopped to think about how we actually, measure it? Today, we're going to crack open the toolkit biologists use to go from just looking at nature to actually calculating evolution.
And that's a fantastic question, right? We can see this incredible variety of life everywhere we look. But how do you turn that into cold, hard data? To really prove a population's genetic makeup is changing, you need a way to quantify it, to actually put numbers on it.
And just so we're clear, this isn't some side quest. The College Board themselves basically says that if you don't get the mechanics of evolution, you're not really doing AP biology. It's the absolute core of the course. So let's get you the tools you need to nail this. Okay, our first tool is a really clever bit of thinking from population genetics. It's called the Hardy-Weinberg equilibrium. I want you to think of it as a perfect, unchanging genetic ruler that we can hold up against the real world to measure change.
Now here's the twist, and this is the really cool part. The Hardy-Weinberg principle describes a population that is not evolving at all. It's completely static. It's basically the control group in a giant experiment. So if we look at a real population and its numbers don't match this perfect baseline, well, that's a huge red flag that evolution is happening. It tells us some force, like natural selection, is pushing things around.
And this whole idea boils down to these two beautiful little equations. The first one, P plus Q equals 1, is just looking at the alleles. The frequency of the dominant allele, that's P, plus the frequency of the recessive one, Q, has to add up to 100%. Simple enough. The second one looks at the actual combinations, the genotypes. So you've got your homozygous dominants, P squared, your heterozygotes, 2PQ, and your homozygous recessives, Q squared. Add all those up, and you've got to get 100% of the population. These two equations are our rulers. All right, enough theory. It's one thing to see the equations, it's another to actually use them. Let's put this ruler to work with a classic AP Bio problem. We're going to head out to a field of sheep. Okay, here's the scenario. We've got a flock of 900 sheep. We know white wool is the dominant trait, and black wool is recessive. Now, we can walk out there and count that exactly 9 of them are black. So the question is, can we take that single piece of information, just the number of black sheep, and figure out the frequency of both alleles in the entire flock? Turns out, yes, we can. And there's a simple game plan. You always start with what you can see, which is the homozygous recessive individuals. That's our Q squared. Once you have that, you can find Q, and once you know Q, well, finding P is a piece of cake. Let's plug in the numbers. Okay, step 1. Let's start with our black sheep. We saw 9 of them in a population of 900. Since black is the recessive trait, we know their genotype has to be homozygous recessive. So the frequency of that genotype, our Q squared, is just 9 divided by 900, and that gives us 0.01. Easy. Now that we have Q squared, finding Q itself, the frequency of the recessive allele in the whole gene pool, is super simple. We just take the square root of 0.01, which is 0.1, and that's it, that's Q. So we now know that 10% of all the alleles for wool color in this entire population are the recessive black wool version. And for the grand finale, we just go back to that first simple equation, P plus Q equals 1. If we know Q is 0.1, well then P has to be 1 minus 0.1, which leaves us with 0.9. So 90% of the alleles floating around in this sheep population code for the dominant, white wool. And boom! Just like that, we took one small, observable fact and used it to paint a complete picture of the population's entire gene pool. The Hardy-Weinberg equations literally let us see the invisible genetic frequencies. Pretty powerful stuff. But what happens when the numbers don't match? What if a population is actually evolving? This is where our genetic ruler turns into a detective's tool. So let's investigate the curious case of some shipwrecked cats. Before we dive into the data, you need to know about something called the founder effect. Imagine a big, diverse population, and just a tiny handful of individuals gets separated, say on an island. Well, just by random chance, the mix of alleles in that small founding group might be very different from the original population. That's a type of genetic drift, which is basically evolution by random chance. So here's the evidence from our cat's drastophy. A few cats end up on an island, and their population grows to 20,000. Using Hardy-Weinberg, we can calculate the numbers we should see if no evolution was happening. That's the expected column. But then we go out and actually count the cats on the island, and we get the observed numbers. And whoa, look at that. They don't match up at all. There are way more cats with folded ears and way more heterozygotes that our perfect ruler predicted. Something's up. So is this difference a big deal, or could it just be a random fluke? To figure that out, scientists use a statistical tool called a chi-square test. You can think of it as a fluke detector, and in this case, our calculated value is a massive 141.6. The critical value, which is kind of like the line for what could be random chance, is only 5.991. Since our number is just astronomically higher, the test screams that this difference is statistically significant. It is absolutely not a fluke. And this is the real magic of this tool. By showing that the population's numbers are way off from the Hardy-Weinberg prediction, we have statistical proof. We have detected evolution in action. That founder effect created a brand new gene pool, and we just measured it. Okay, so Hardy-Weinberg is amazing for measuring evolution happening in real time. But what about the big picture? How do we map relationships that stretch back millions and millions of years? For that, we need a totally different tool, one that lets us build a family tree for all of life. The challenge here is completely different, right? We're not tracking allele frequencies anymore. We're hunting for clues hidden deep inside molecules, like proteins and DNA, that whisper stories about who's related to whom across these vast oceans of time. And this is how it works. On the left, you've got this table of raw data. It's just a count of how many amino acid differences there are in one specific protein across a few different animals. Biologists then take that data and feed it into a program that spits out the image on the right, a phylogenetic tree. It's an evolutionary map, and its entire structure is built directly from that molecular data. The idea behind this is so simple and so elegant. Think of it like this. When two species split from a common ancestor, their DNA starts to accumulate random changes, or mutations, over time. If these changes happen at a more or less steady rate, they act like a molecular clock. So the more differences you see between two species, the more time has passed since they split. Fewer differences means they're close cousins. Tons of differences means they're very, very distant relatives. So we have these two amazing tools, right? One for taking a snapshot of evolution right now, and one for mapping its history. Let's wrap things up by talking about why the kind of evidence you use is so important in biology. Sure, you could try to build that family tree using physical traits, what scientists call morphology, but that can get really tricky. Sometimes two totally unrelated species will evolve similar-looking parts because they live in similar ways. Think about the wings of a bat and the wings of a bee. They look similar, but they have completely different origins. That's called convergent evolution, and it can really throw you off. But molecular data, it cuts right through that noise. It shows you the actual genetic history, which makes it a way, way more reliable tool for drawing these family trees. And that kind of leaves us with one last big thought. We've used these tools to figure out the stories of sheep, cats, and even ancient animals. It just makes you wonder, right? What other amazing stories of survival, of change, of journeys across the globe are written in the DNA of every living thing, just sitting there, waiting for us to find the right tools to read them?